I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the Hudson River, down by the river, that's where I want to be. You may be feeling cramped and crowded. You may be feeling restless and bored. I know a place in your wide ails will leave you refreshed with your mind restored. I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the Hudson River, down by the river, that's where I want to be. You never know what you might find there. You never know what the tide will bring. But I know you'll find peace of mind there. Something about it makes me wanna sing. I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the mighty Hudson, down by the river, that's where I wanna be. Just make your way down to the water. That river will bring the world to your feet. Might be something washed down from the mountains or dredged right out from the ocean deep. I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the Hudson River, down by the river, that's where I want to be. Once we got careless about our river, let it fill up with pollution and trash. Now we're trying hard to protect it, want to make sure that our river will last. I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the Hudson River, down by the river, that's where I want to be. I'll meet you down by the river, down by the river, down by the river, to see what we can see. Down by the river, by the Hudson River, down by the river, that's where I want to be. Down by the river, that's where I want to be. Down by the river, that's where I want to be. Down by the river, that's where I want to be.
Well, good evening, everyone. I'm Steve Stan. I'm the board president of Clearwater and the one of two acting executive directors at Clearwater at the moment. And I'd like to welcome you all to the fourth and final webinar in our series celebrating the release of The Hudson, an illustrated guide to the living river from the Rutgers University Press. This is a Clearwater project that got started almost 30 years ago now. And the webinar is featuring a number of different presenters who have worked on the book and whose work has been vital to the writing of the book. Tonight, we're going to hear from Dr. David Strayer, the Carey Institute of Ecosystem Studies, with a few thoughts about science and the future of the Hudson River. And I'll introduce Dave in a few minutes. A bit about the book first. It first came out in 1996. It was a response to the many calls we got at Clearwater asking for information about this subject or that subject. And although much of the information existed in scientific papers or in popular press articles or occasionally in a book, there wasn't one place where you could go to find a bunch of information that would be useful to a teacher or a parent helping a child study the river. So we were fortunate enough to be able to get some grant funding to put together this book. It first appeared in 1996 from the Rutgers University Press. A second edition came out in 2006. And given the change in environmental issues over the years and increased understanding of the river and its history, third edition was merited publication, and it has just come out. You can find more information about it on the Clearwater website if you'd like to order a copy. The co-authors of the book are, besides myself, Roger Panetta, historian with Fordham University, Brian Forrest, a former Clearwater educator, now at Indiana University, and Maya Nemisto, another former Clearwater educator who is working with the Hudson River Estuary Program and the Hudson River National Estuarine Research Reserve right now. We had originally planned to have all of the co-authors present as part of this series. However, Maya was unable to do so, and we were very lucky to be able to have Dave Strayer join us, and you'll be hearing from him tonight and learning more about why we're so happy to have him. A book like the Hudson really depends on many more people than just the authors, and I'd like to recognize many of those folks now. First, the scientists, historians, and other researchers whose work really forms the foundation of the book. And also the designers, the illustrators, the photographers, the collectors, and institutions who provided the art. One of the differences between this volume and the previous ones is that this one is largely in color. Some of the previous black and white illustrations, the drawings and graphs are still in black and white, but the photographs are largely now in color, a great improvement. We need to thank the Rutgers staff and editors, but also the many folks at Clearwater, at the Hudson River Estuary Program, at the Carey Institute, at many other institutions who helped us with advice and review and other assistance in putting this book together. And finally, it wouldn't have happened without the funders, grant makers, especially the Hudson River Foundation, which provided a substantial grant to get the project started and has been supportive ever since, but also Clearwater members who, by their contributions, helped to pay the salaries of the folks who put together the book. Well, tonight's presenter is Dr. David Strayer. He's a distinguished senior scientist emeritus at the Carey Institute of Ecosystem Studies, a world-renowned environmental research institute in Millbrook, New York, in the Hudson Valley. He's been based there since 1983, and he's a freshwater ecologist, and much of his work is focused on measuring the long-term effects of zebra mussels on the Hudson River. He has a particular interest in mollusks. And he has helped to create one of the longest and most detailed case studies of invasion ecology. He's also an author. He wrote a book called The Hudson Primer, The Ecology of an Iconic River. And for those of you who are coming to the Hudson through the book being celebrated tonight, this is a great next step. It really focuses on ecology of the river and presents it in a very readable and understandable way. For those of you who are interested in diving deeper into the science, I highly recommend a paper for which Dave was the lead author, but he was joined by many of his colleagues at the Carey Institute in putting together. It's called Decadal Scale Change in a Large River Ecosystem. It was published in Bioscience in 2014. And it's a great summation of some of the changes in the river over time. For those, we found that it was invaluable for rewriting and preparing the third edition of the Hudson. And I will put a link to that paper. It is available online. I'll put a link to it in the chat once we start on the webinar tonight. Dr. Strayer is also past president of the Society for Freshwater Science, and he works on a lot of broader issues in freshwater conservation ecology, and particularly in invasion ecology. And I'm sure you'll hear something about that tonight. So with that, Dave, I'm going to turn it over to you. Thanks for being here.
Thank you, Steve. I'm very happy to be here tonight. And I'm hoping Can you, is that up now, Steve? Can you see that and hear me okay? So I want to talk a little bit tonight about what science has taught us in the last few years about how the Hudson River works. And then I want to talk about how science can help us navigate the future of the Hudson River and the challenges we're going to be facing. And right at the beginning, I need to emphasize that the work I'm going to be talking about tonight comes from a large community of researchers on the Hudson River. Here are a few of the pictures of some of the people that I could find readily, but there are literally hundreds of people who've worked to gather this information together. And my own work is only a small, little, tiny bit of what I'll be talking about tonight. I want to open by asking the hard question that the authors have been afraid to ask. Why do we need this book? Sure, it's an attractively priced book. It's well written. It's full of color illustrations. It's a nice book, but we have lots of books about the ecology of aquatic ecosystems. Have been around for a long time. This Coker book, for example, 1954, is a book I knew about when I was a young man. That was a long time ago indeed. And it explained how systems like the Hudson River work. So why do we need a new book? And in fact, as long as I'm feeling grumpy, why do we need to keep doing this scientific research? I mean, if we already knew by 1954 kind of how aquatic ecosystems worked, this looks like a lot of work going on the river and lifting these heavy things and then going back in the lab and analyzing the samples. Why do we need to keep doing scientific research on the Hudson? So if you went to a book like Coker or any other book, by the 1970s, we had this general idea about how aquatic ecosystems work. And it went something like this, that nutrients came in from the watershed into the river or lake. And by nutrients, I mean nitrogen or phosphorus, fertilizer. And that fueled phytoplankton growth. The more nutrients you put into the ecosystem, the greener the lake or river would be. Or if you wanted to try to make the lake or river a little clearer, you could cut back on the nutrient inputs a little bit to make it clearer. The phytoplankton, in turn, is the base of the food web. It feeds zooplankton down here in the center of the picture. I hope you can see my little arrow here. And the zooplankton, in turn, are fish food. In a river like the Hudson, a lot of the fish are anadromous. They're migratory. The adults live out in the ocean. Fish like the river herrings and the shad. The adults come into the river in the springtime. They lay their eggs there. The river fills up with immense numbers of young shad and herring during the summertime. When fall comes, they drift back to the ocean and get fat in the ocean for a few years before returning to the Hudson. And in the shallows of the river, there are aquatic plants. And one of the things those aquatic plants do is put dissolved oxygen into water and make it suitable for fish life, for example. And all this, as I said, was well known by the 70s. The trouble is that almost none of what I just said really applies very well to the Hudson River. And I want to walk you through what we have learned about how the Hudson really does work and how it differs from this sort of simple textbook diagram. The first thing is phytoplankton are not nutrient limited in the Hudson River at most times and places. The graph on the left shows the situation at a place like Poughkeepsie, say where the walkway over the Hudson goes over the river. Steve already mentioned this in his talk. The river there's about 60 feet deep, maybe a little bit more, and it's quite turbid. So the graph here, the vertical axis, here is water depth. So here's the surface of the river up here at 0. Here's the bottom of the river down at 20. The amount of light reaching at a given depth is on the horizontal axis here, 100% of sunlight at the top. And you can see that it gets dark really fast. There's almost no light left by the time you get a quarter of the way down. By the time you get halfway to the bottom of the river, it's about as, if you go out on a fine summer day with a lot of sunlight, halfway down the river, it's about as dark as night there. And down at the bottom of the river, it's darker than any place that most of us will ever be. Now, this presents a problem for the phytoplankton because the river at Poughkeepsie is not stratified. And so that means that when the tidal currents move the water around, the water goes all the way from top to bottom, carrying the phytoplankton with it. They spend a little time up in the top where they get a little bit of light, and then they spend most of their time in the dark. It's almost as if you were living on a conveyor belt that went past the cafeteria maybe every week, once a week. So the phytoplankton have trouble with light more than to do with nutrients. The second thing we know, sorry, I'm having a little trouble here, about the Hudson is that it's just full of nutrients. This shows the amount of input, the annual input of nitrogen on the left and phosphorus on the right in the Hudson. In the old days, in pre-Columbian days, there was just a little bit of nitrogen and phosphorus coming into the river, like shown by the blue bars here. Now we have immense amounts of both nitrogen and phosphorus coming in the red bars. This is the nutrient input rates into the Hudson are by far the highest of any large estuary in the United States. These nutrients are coming in from sewage, they're coming in from lawn fertilizer, from farm fertilizer. And in the case of nitrogen, they're coming in the form of polluted rainwater. What this means is there's always plenty of nutrients for the phytoplankton to grow. So the first thing we can cross off on our little simplified textbook diagram is the phytoplankton in the Hudson are not generally limited by nutrients. Instead, they're limited by light or in some cases by grazing. And as it turns out, the phytoplankton aren't primarily or solely what feeds the zooplankton either. And there are several ways that we know that. The first is a simple budget. This table shows the amount of food material that's coming in at the base of the food web from different sources. And they're all in the same units. So you can see that phytoplankton production contributes 12 to 50 grams carbon per square meter per year. The units don't matter. And about the same amount comes in the primary production of rooted plants. Those inputs are dwarfed by the food that comes in from the watershed. These are little bits of leaves and things that wash in from the watershed, or sort of a dilute tea that's formed when rainwater moves through the soils of the watershed. About 90% of the food coming into the river comes in from the watershed. Only a little bit comes from phytoplankton. If there are any phytoplankton fans in the audience tonight, you might be protesting that the phytoplankton production is good food. This is juicy stuff that the animals like to eat. And the stuff that's coming from the watershed is crummy. And that's true. And so you could argue that maybe the phytoplankton really still is important in supporting the food web, but we have other bits of evidence that show that this stuff coming in from the watershed is also very important. Now, don't freak out. Here's a graph. Steve tells me some of you don't like graphs, and so let's not look at the graph for a minute. Let's just do a little thought experiment. Suppose I've got a greenhouse and it's full with luxuriant vegetation and it's a bright summer day, and the greenhouse is closed. So what happens on that sunny day? We know that the plants are photosynthesizing. They're taking up carbon dioxide from the air in the greenhouse, and they're producing oxygen. So that if we were to measure the oxygen and carbon dioxide in that greenhouse, there'd be more oxygen, less carbon dioxide than the air outside. If I had an exactly identical greenhouse and it was filled up with rabbits and no plants, just bunny rabbits, and I was bringing in rabbit food to feed them, and we watched gas dynamics in that greenhouse, we would see that the rabbits would produce carbon dioxide and they would consume oxygen. So if I just showed you the gas dynamics in those two greenhouses, you could tell me easily which one contained rabbits and which one contained luxuriant vegetation, right? The vegetation would have a lot of oxygen in it. The rabbit greenhouse would have a lot of carbon dioxide. So this graph now shows what's happening in the Hudson with gas dynamics. Think of the Hudson as sort of similar to that greenhouse I was just talking about. And what you can see, this is on the x-axis now is time, this is several years. On the y-axis is the amount of carbon dioxide in the water and the amount of oxygen in the water. The red horizontal reference lines shows how much carbon dioxide and oxygen is in the atmosphere. And you can see that nearly always there's lots more carbon dioxide in the Hudson and lots less oxygen in the Hudson than in the atmosphere. And this tells us that the Hudson acts more like the greenhouse that's full of rabbits. It is consuming food that is brought in from outside. And then finally, this is just so cool, I have to show this to you. We have even more direct evidence the food web is supported by material coming in from the watershed, and not just any material coming in from the watershed, but truly ancient material that was produced in the watershed a long time ago. And to do this, we need to look at the carbon isotopes of plants and animals in the river. Now, the element carbon exists in 3 isotopes. So there's carbon 12, that's ordinary carbon with 6 neutrons and 6 protons. And about 99% of the carbon in the world is carbon 12. There's carbon 13, which has an extra neutron, it's a little bit heavier, and about 1% of the carbon in the world is carbon 13. And then there's just an eensy-beensy tiny little bit of carbon 14, which is radioactive. I think many of you will have heard of carbon 14. And the cool thing about carbon, the useful thing about carbon, is you are what you eat with respect to carbon. So if I ate a diet that was 5% carbon 13, eventually I would become 5% carbon 13. So you can look at the carbon isotope content of various kinds of foods and the animals that eat them and determine what foods the animal is eating. So the animal we're studying here is this common zooplankton. This is Bosmina. It's a little less than 1 millimeter long. It's an important fish food, and the river just fills up with these things in late spring. So the question is, what are these guys eating? And so what Nina Caraco and her group did was they looked at the carbon isotope content of several possible kinds of Bosmina food. So we've got carbon 14 on the vertical axis, carbon 13 on the horizontal axis, phytoplankton here, I hope you can see this on the left, floating leaved aquatic vegetation, that's water chestnut. And on the right here, SAV, submerged aquatic vegetation, that's wild celery. And then you also have leaves from trees right now in the watershed, modern terrestrial. And so if Bosmina is eating those, if you grind up a Bosmina and measure its carbon isotope content, it should fall somewhere around this green triangle, right? But what you see instead is that Bosmina, labeled zooplankton here, is way down in the bottom of the diagram. The only other thing that could be feeding it is aged terrestrial matter. This is material that was produced by trees and vegetation in the watershed a long, long time ago. The average age of this stuff in this diagram is 2,000 years. So this is material that was produced on a fine summer day 2,000 years ago when Jesus was a little boy. And it sat around in the soils of the watershed uneaten because it was so crummy. And then it finally washes into the river after thousands of years. And then it turns out that that is what the zooplankton are feeding on. They're feeding about half on phytoplankton and other modern material in the river, and about half on this very ancient material from the watershed. So all of these tedious lines of evidence that I've just gone through tell us that the river's food web is not solely or even primarily supported by phytoplankton, but it's supported by other things as well. The next thing that's not true about the Hudson that's shown in this diagram is that the fish eat zooplankton primarily. There have been lots of studies in the last 20 or 30 or 40 years on what fish in the Hudson eat. And I've arrayed the fish species here by what they eat according to plankton versus benthos. The benthos are the animals that live at the bottom of the river, like insects and shellfish and crustaceans. And you can see that there's only one fish, this beautiful bay anchovy, that feeds primarily on, or really wholly on phytoplankton, or on zooplankton. The shad and herring are about 50-50, And then there's a whole bunch of fish species in the Hudson that feed primarily or solely on benthic animals, on sediment-dwelling animals. So it's not true that the fish in the Hudson are fed primarily by zooplankton, like in this diagram. Nor is it true that fish migration occurs exactly as in the textbooks. And this is another example that is technically very cool, which I'm going to need to explain for a second. This is from Karin Lindberg's lab up at SUNY ESF. Fish have got these ear bones. They're called otoliths. This is a little tiny bone up on the fish's head. There's where the otolith is in the fish's head. And if you look closely at the otolith, it looks like the cross section of a tree, and for the same reasons. The rings in this otolith, lost my arrow here. The rings in the otolith are growth rings. And just like the rings in a tree represent years, in this case, the little tiny rings represent days in the fish's existence. So this part of the otolith was built when the fish was a little baby, and as it grew up, the otolith grew bigger and bigger and bigger. And this is the last day of the fish's life before Karin caught it. It turns out that Karin knows how to analyze the chemistry of these otoliths. They take a laser beam and they shine it at the otolith, and that laser vaporizes the chemicals in the otolith, which are then sucked into a sophisticated instrument for analyzing chemistry. And by knowing the chemistry of the otolith, they can know where the fish was. So if you look at the bottom center picture here, this is SR map. SR is the element strontium. So the darker colors show parts of the otolith that have more strontium in them. Strontium is much more abundant in the ocean than it is in the Hudson River. So what you see in this case is this little fish, when it was small, lived in the Hudson River where there wasn't much strontium. And then it moved out at the end of its first year into the ocean. So that's what it was supposed to do. But it turns out when Karin and her group analyzed the ear bones of many individual, in this case it's river herring, they found that many of the fish had not read the textbooks, even though I don't understand, fish are supposed to spend a lot of time in school, but apparently they didn't learn this part. So about 70% of these river herrings came back into the river when they were 1 year old. So they spent not just their first year, but their second year as well in the Hudson. About 20% of the fish don't go all the way to the ocean and the river, but in the winter, but they spend the winter in the river. And so what this tells us is that textbook description of fish migration isn't incorrect, but that the actual fish, individual fish, are much more variable. There's much more flexibility and patterns of migration than the textbook would suggest. So that isn't right either. And then this is my last gruesome graph. So please, I'll try to walk you through this. Not all aquatic plants add oxygen to the water. In Hudson, there are two common kinds of aquatic plants, which if you've been on the river, you've seen them. You've got the native water celery here, and sometimes this is called eelgrass or just grass. There are huge beds of this stuff all through the shallows in the freshwater part of the river. This is a native species. On the right is the water chestnut, which is a non-native species. It was introduced deliberately in the 19th century as an ornamental. It's a really, really tough plant to go through. I don't know if any of you tried to swim or wade or take a boat through this stuff, but it's just impenetrable. You get some sense of that from the picture of Nina and Heather here in a water chestnut bed. So the diagram at the bottom shows the difference in what happens in wild celery beds in green and water chestnut beds in orange. The horizontal axis here is time. It's 3 days, 3 fine summer days. I think it's July, I believe. And you can see at the top, night in gray, day in yellow. So night, day, and night, day, and so forth. You can also see the tidal cycle here. This gray line at the bottom shows water depth at the study site. And you can see here's a flood, the water comes in, and then it reverses down to an ebb. Now, oxygen dynamics in the wild celery bed in green here are what you would expect a respectable aquatic plant to do. The light comes on and it starts photosynthesizing, making oxygen, and the amount of oxygen in the water rises and rises and rises until just before sunset, and then oxygen falls and it falls, and then the lights come back on and the oxygen goes up again. In the water chestnut bed, something very different happens. And I'm going to explain why this happens before I walk you through what happens. This canopy, this bed of water chestnuts is so dense that if you take a light meter and you put it in the water beneath the water chestnut on a day like this day in the picture here, it's as dark as night in the water underneath that water chestnut. That means there's no photosynthesis occurring in the water under the water chestnut leaves. Instead, there's a lot of respiration from the water chestnut stems and roots and all of the animals and bacteria that live in the mud underneath the bed. This plant produces, water chestnut produces oxygen, lots of it, but the oxygen is released into the air and not the water. So what happens to oxygen concentrations in water chestnut beds is shown in the orange lines here. The thin line shows the very edge of the water chestnut bed. The thick line shows what happens deep inside the water chestnut bed. You can see that the flood tide brings in water from the river with a high oxygen content, and then that respiration eats up all the oxygen out of the water. In a period of just a few hours, this is 6 hours here, right, from flood to ebb. In some cases, all of the oxygen in the water, all of it is stripped from the water. Now, zero oxygen is a problem for most animals like us and like most fish and most invertebrates. They need oxygen. And so these water chestnut beds are an inhospitable habitat for many animals. I'm not going to talk about it now, but there's also some really interesting and important microbial processes that occur in these water chestnut beds because there's lots of juicy food, lots of juicy organic matter, and no oxygen. So these are really unusual environments in the water chestnut beds, and they are unusual because water chestnuts don't act like a respectable aquatic plant. They don't add oxygen to the water. Instead, they strip oxygen from the water. So here's our little scorecard at this point. Pretty much everything I said that a standard aquatic ecology textbook would say is either wrong or you'd have to qualify the statement if you were talking about the Hudson. So why is that interesting? Well, it seems to me that if you want to manage an ecosystem, you want to understand how it works before you try to manage it. This diagram here is the wiring diagram for a motorcycle. And if I were a mechanic, I would want to be sure I had the right schematic before I started working on a motorcycle. And I think that what the science has taught us in the last few decades about the Hudson is that we need to have the right schematic if we're going to manage the Hudson wisely. And that schematic for the Hudson is not the same as it would be in the textbooks. So I think I've answered the one question. I think we kind of do need science. Why do we need a textbook? Steve actually said something about this in his intro tonight. The reason we need the textbook is because a lot of people who care about the river, like you guys, and managers and policymakers, they don't want to read this stuff. This on the left here, this is an actual scientific paper. You guys don't want to read this. I don't want to read this. Instead, we have this really nice, well-written, well-illustrated, and attractively priced new textbook that explains how the river works in terms that people can understand and use. All right. The other reason we need science, I think, is that the river is changing. The Hudson River is changing. When you put up a diagram like this textbook food web or like the wiring diagram for a motorcycle, you're assuming that it's the same over time. The ecosystem works the same in 1970 as it did in 2000 as it's going to work in 2050. Nobody's going to sneak into your shop in the middle of the night and rewire the motorcycle without telling you. The Hudson's changing though. And so it's as if every year the wiring diagram for the Hudson is changing. And I want to give you just a couple of examples to, to explain what I mean and to try to convince you that the Hudson really is changing. The first is the biological composition. The species that live in the river change over time, change pretty rapidly, actually. On the left here, this graph shows the number of non-native species that we know of that are established in the wild in the fresh waters of the Hudson River basins. So this is not just the estuary, but it's the tributaries as well. You can see a couple things about this. The first is, if you look at the scaling on the vertical axis, 125, I think the last estimate's 122 species. That's a lot of new species that weren't in our area in 1600. You can also see that the line looks pretty straight for ecology. That's really straight. And that means that the rate of detection of new invaders has been constant as far back as we can see, about 1850. And that rate is, we see about 6 or 7 new species every decade in the basin. And about one of those species, about one species a decade, has strong ecological impacts. So on the right here are there photographs or pictures of some of the invaders that have appeared in the Hudson since I started working on the river about 1990. Zebra mussels, the aquatic plant hydrilla, channel catfish, Chinese mitten crabs, and freshwater drum or sheephead. These are all species that are capable of having strong impacts on the river, and none of them were in the river when I started working on it. So I said some of these species can have strong ecological impacts. I have, in fact, delivered whole lectures just on the impacts of these species. I'm just going to show you a couple bits of data from the zebra mussel invasion to show you just how large and far-reaching those changes can be. This is the summary of work that Carey and our partners have done on the effects of the zebra mussel in the first 5 or 10 years after it arrived. It appeared in 1991. It was super abundant by 1992, and these are the effects going up to about the year 2000, more or less. Each box shows a different part of the ecosystem. The size of the box is proportional to the change that zebra mussels caused. So big boxes are things that zebra mussels cause to increase. Little boxes are things they cause to decrease. I've also color-coded these, so red is a decline, just like it is on your balance sheet, and blue is an increase. So you can see that zebra mussels ate about 80% of the phytoplankton. The amount of phytoplankton in the river fell by about 80%, despite what I said about other things being important in the food web. Phytoplankton is important in the food web, and we saw a decline in everything in the river that depends on phytoplankton. So this is a sort of withering away of the open water food web in the Hudson River. At the same time, the loss of phytoplankton meant the river got a little bit clearer, only about 25% clearer, but that was enough to improve growing conditions for those rooted aquatic plants like wild celery. So we saw an increase in the shallow water part of the food web, more plant growth, more invertebrates and fish living in those plant beds. You might say, looking at this diagram, that the zebra mussel was kind of a wash what they did in the Hudson River. We had an increase on one hand in the shallow water and a decrease in the open deep water of the river. But there's a lot more area in the deep water than there is in the shallow. So on a whole, there was a general diminishment in the upper parts of the food web as a result of zebra mussel invasion. Now, the reason I qualified my statement by saying that these data are from the first 10 years or so of the invasion is that the zebra mussel population in the river and its effects are still changing. When an invasion occurs, it doesn't instantaneously change the river in a way and then just stay that in a changed state. Instead, the invader and the ecosystem spend some time accommodating to one another. And I just want to show you one example of this. So The left graph is the abundance of large zebra mussels. These are about an inch long, about 2-year-old. And so what you can see is about the year 2005, they disappeared from the river. What's happening is the blue crabs that come up the river every summer discovered zebra mussels and they started eating them. And so there's just as many zebra mussels as there ever was, but they're just not big because the crabs eat them before they have a chance to get big. These are the abundance of a small zooplankton, a kind of small zooplankton called rotifers. Little tiny fish eat these. And you can see at the same time that the big zebra mussels disappeared from the river, the rotifers reappeared back towards their pre-invasion densities. And we think this is because little zebra mussels can't eat rotifers. Another really interesting example of surprising change in the river is the rapid evolution that's been occurring. This is rapid evolution that is mostly occurred as a result of selective pressures imposed by bad pollution of the Hudson. There are a couple of examples of this. Brian, I think, mentioned this last week a little bit, and this concerns the tomcod. They're called frost fish. Sometimes these are fish small codfish, less than a foot long, that live in the brackish parts of the Hudson River. They're residents there. And the distressing thing about these is that many of them have liver cancers that kill them. Ike Wergen at NYU has done a lot of work on these fish, and it turns out that some of the kinds of pollution that cause these liver cancers are things like PCBs and dioxin that were released from various industrial facilities along the course of the river. Ike also found, though, that in the last 50 to 100 years, there's been dramatic evolution in the tomcod population so that they're now far less sensitive to these pollutants than tomcod in nearby rivers. There's a gene here called AHR2, and it has 2 alleles. One in black here was probably dominant all up and down the East Coast in the old days, and it's highly sensitive. Fish with this allele are highly sensitive to pollutants. The white allele has recently increased probably in the Hudson River, and this genotype is less sensitive to pollution. So the fish have evolved. We often think of evolution as something that occurred in the distant past and occurs slowly over millions of years, but evolution is happening in our lifetimes in the Hudson River, changing the character of the species that occur in the river. All right, so that gives us another answer about why we need new science and new textbooks. The old stuff goes out of date because the ecosystem changes. All right. This change has been occurring ever since Europeans came on the scene, maybe well before that, in fact. And this is our attempt to indicate some of the factors responsible for broad-scale change in the Hudson since 1750. And you can see that the actors have changed from deforestation, oyster overharvest in the early history of the river to human-caused climate change in more recent years. But the point I'm trying to make here is that the Hudson has been changing for at least 250 years, dramatically changing, to the point that if you were to ask an old guy at any point in this period whether what the Hudson was like when he was a kid, he or she was a kid, they would say, well, you know, it's not the same river as when I was a kid. And there's every reason to think that this change is going to continue into the future and perhaps even accelerate. So I'm going to close by considering briefly what kinds of change we might expect to see in the Hudson in the next few decades, and what role science might play in helping us understand and manage that change. I'm not as smart as this guy in the picture here, so I don't know exactly what kinds of changes are going to occur. But I'm pretty sure we're going to see at least 3 classes of changes being important in the river. Deliberate management of the river, things that we know are coming but aren't controlling very well, And then finally, surprises. I want to say a few words about each of these. Deliberate management. Government agencies and nonprofits now routinely manage the river to try to provide more benefit to all of us. And examples of what I'm talking about are fisheries management. We have catch limits, harvest limits on fish. We're trying to rehabilitate some of the toxics out of the river. The most famous case is the big PCB dredging project in the upper river. And increasingly in the last few years, we're trying to restore habitats, degraded habitats, to provide better ecological value. So the really exciting thing about this is we know from past deliberate management, this deliberate management can succeed. And I'll remind you with a couple examples you may already know. The picture on the left is Foundry Cove down near Cold Spring. This was the site of a battery factory for a long time. It was spectacularly contaminated. The sediments of this cove were up to 25% metals, cadmium and nickel, by weight. And cadmium especially is a problematic chemical. It's toxic, and it was bleeding out into the river. So a few years ago, this Foundry Cove was remediated. They took out a lot of the metals. They put clean cap material on the top. And this is what Foundry Cove looks like after the remediation. And we know that there's a lot less cadmium going into the river from Foundry Cove now. So it was expensive, it was complicated, but it improved the conditions there. These are striped bass on the right. They were in trouble coast-wide, all up and down the coast in the 70s and early 80s. Stocks were declining. The eastern states all got together and imposed management regimes, and the striped bass populations came back. So we know this sort of thing can work. These management actions, I think, need to be monitored. This is a nice example of some really positive work that's occurring up and down the Hudson now to try to improve the quality of shorelines. The left-hand side shows a failing bulkhead. This is also, to an ecologist's eye, that this is a site of poor ecological value. On the right is what they did with the site afterwards. This looks a lot better. We need to monitor to see how well this shoreline treatment persists into the future, or whether it falls apart, and how much better ecological value it provides as we hypothesize. So what science can do here is it can help us all identify the best opportunities for management and set goals. And I think we need to work hard to treat management as an experiment, because I already tried to convince you the Hudson is complicated and changing. And so we don't actually know always how well these management efforts will work. So I would argue that we need to design projects so they can be evaluated, then go out and actually evaluate them by collecting information on project performance and use that information to improve future management. And that may seem obvious, but many times all of this evaluation stuff gets cut because budgets are too small for it. And so people say, well, we'll just build the project and hope for the best. The fisheries managers are doing a good job here, and the rest of us maybe need to do a little better. The second class of things I want to talk about are, I don't have a good term for these. I'm calling them poorly controlled drivers. These are things that we know are coming. They're going to affect the river. It's too late to prevent them. And the two examples that are obvious to me are climate change and continued invasion of non-native species. Even if tomorrow we were to do all the things to prevent future climate change, and all the things to prevent future invasions of non-native species, we would still get changes in the climate and in more species for the next few decades. We're already committed to that. So I want to say a few words about each of those factors. And the other thing they have in common is that both of climate change and non-native species invasions can have really large, far-reaching impacts on the river that are hard to predict in detail. So there's four aspects of climate change I want to mention. I think this is familiar to a lot of you. Temperatures are rising. The graph on the upper right shows the temperatures are already rising in the 20th century, and the projections to the right of the dashed line there show accelerated warming in the coming decades. So this rising temperatures are going to have so many effects on the river. The obvious ones are going to be the shifts in timing of seasonal events. You know, already we're seeing birds migrating earlier, plants flowering earlier. We have evidence that that it's affecting the timing of when fish come up the river now and spawn in the spring. There's also some disturbing uh, suggestions that we're starting to lose some of the cold-loving fish species from the river. The smelt are pretty much gone, and the tomcod are struggling down in the lower river. These are fish that really do better further north, and as the Hudson warms, they're going to have some trouble. Another pervasive impact of climate change is going to be rising sea level. This is going to affect the Hudson all the way up to the foot of the dam at Troy, and we're already seeing property damage. We're going to see more property damage, and we're going to see drowning out of some of the valuable shallow water habitats in the river, and we're already seeing these things. And again, the graph on the lower right now shows that sea level has been rising through the 20th century, and the projections are uncertain in the 21st, but they show a wide range of possible scenarios, some of a very great sea level rise indeed. The other effects that we can expect from climate change are more frequent severe storms. The picture in the right, we're going to see that sort of thing more often. These severe storms damage not just infrastructure, but also the ecological habitats. The graph on the lower right is the number of places that volunteers looked for submerged vegetation and found it over a course of time. This is a group, Stuart Finley's group, that's been collecting these data. The red arrow shows Irene and Lee, a couple of big storms a few years ago. And what you can see is the year after Irene, there were a lot fewer aquatic plants in the river. This is because the sediment brought in by the storms probably buried the plant beds. And it took several years for these plant beds to recover back to where they were before the storms. The other aspect of climate change that people don't always think about right away is that the human responses to climate change may themselves cause ecological damage, especially if they're conceived in crisis, which alas is often the way we do these things. So if we get a lot of storm damage, there may be attempts to reinforce shorelines hastily, to raise rail lines hastily, and especially if these are done hastily, we can expect some adverse ecological impacts. I think I already showed you this graph. We're going to have more invasions. We still aren't controlling invasions. The pictures on the right show a bunch of species that are already established in areas nearby the Hudson and are very likely to appear in the Hudson in the coming years, even if we stop all invasions to North America tomorrow. So from the top, it's a snakehead, the other fish, flathead catfish, one of the Asian carps, New Zealand mud snail. This is a amphipod from the Caspian, and a bloody red shrimp on the lower right. These are all species that are very likely going to appear in the river. So what can we do about this second class of factors? Well, we know we're going to see effects now. So in the short term, I guess we can track them. We can use science to track what the river is really doing in response to these factors, and we can mitigate effects where we can. So this might be raising fish in a hatchery, planting out plants after a big storm, these sorts of things. Over the longer term, I think we need to do a better job using scientific information to drive policy. And I don't know exactly how to do this. We're now finally stepping up to confront climate change about literally 30 years after we had the science to support those policy changes, and we're still not controlling new invasions. I think part of the challenge is that we need to figure out how better to think locally and act globally. The people in the Hudson Valley have done a really, really admirable job confronting local environmental problems with local action. But things like climate change and invasions are examples of of drivers that I don't think can easily be solved solely by local action. And there are more of these coming down the pike. Things like synthetic biology, where this just scares the bejesus out of me, but they're talking about like high school kids being able to make new life forms in a few years. That sounds like a wonderful idea to me. And climate engineering releases new chemicals in the environment. It would be really lovely if we didn't have to wait for 30 or 40 years after science was around to be able to tackle these issues. The last thing I want to talk about is surprises. If you were to go back to 1970 or 1980 or something like that and ask people who cared about the Hudson, knew about the Hudson, what the big issues were, I guess they probably would have said water pollution and overharvest of valuable species. I don't think any of the issues that I list here on the left side would have been front and center in 1970 or 1980. We just didn't expect them. I don't think there's any reason to think we won't be surprised in the future. I've been a little disappointed how many times when I've been in planning exercises where people seem to assume that they can think of everything that's going to happen in the future, and that the reason people were surprised in the past is they were stupid. And those guys back in 1970 and 1980 weren't that stupid. The world's complicated and hard to predict. And I think it's safe to assume that we're going to be confronted by some surprises in the Hudson. So what do we do about it in particular? What does science do about it? Well, we can be alert to watch for things changing in the river. This means scientific monitoring programs. It also means engaging citizenry, because there's so many more regular people out on the river every day watching for things than there are scientists. I'm concerned about this partly because we're losing some existing monitoring programs right now in the river. And the last thing we need right now in the Hudson is less information about how the river is doing. We need to be prepared to jump on these surprises when they occur and study them. We're really lucky in the Hudson Valley. There's some really great scientific capacity there. There are a lot of good research groups there. We need to maintain that. I don't know what to tell you about that, except that at the federal level, the big science funder is the National Science Foundation, which amazingly has been largely free of political influence and supports it keeps our science going. It keeps that capacity going. So if you ever meet a congressperson and you want to put in a good word for the National Science Foundation, that would be just great. And then we have to be prepared to have management structures that can shift course by using new scientific information on these surprises. All right, finally, my conclusions. I hope I've been able to convince you in the last 45 minutes or so that science has learned a lot in the last few decades about how the Hudson actually works, and it's not always like the textbooks. I think science is going to be vital in addressing the challenges of the changing Hudson, that we know the Hudson will be changing. We know that's going to challenge us in the future. And I think science has to be part of that team that addresses those challenges. Part is a key point. Science is not going to do this alone. We've got to be able to use that scientific information. So scientists have to work closely with a whole bunch of other people. I didn't list enough people on here. There's a whole bunch of other people that need to be part of this future of the Hudson. And this is one reason why I'm particularly happy to be able to talk to all of you tonight, because I'm a scientist and I expect a lot of you are not, and that's awesome. And I'm especially glad to see the third edition of this great book appearing, because that is also a part of this solution. And I guess all of you probably believe this or you wouldn't be on the call tonight, that the future of the Hudson is in our hands, and it depends how well we apply our skills and energy to the challenges we face. So if there's time, I'd be happy to discuss things with you all or take questions.
Well, thanks so much, Dave. We do have a bunch of questions. So we'll start right in with that. One thing I wanted to do before we get into this week's questions is have you respond to a couple of questions that we had last week, specifically about zebra mussels. And given your expertise regarding zebra mussels, we thought we'd start with those. One was, well, One was, can you eat them? And what eats them? And I think you sort of answered the one of those questions tonight, but I'll start with that first one. Would you eat them?
Thank you for the reminder, Steve. I personally would not eat them. They're edible. They would be wholesome to eat. And if you're a shellfish lover and you're curious, you're welcome to try some zebra mussels. There are two important cautions to that. The first is you want to get them from clean water. And many of you probably know that shellfish are very good at picking up contaminants out of the water where they feed. So, you know, get your zebra mussels out of clean water and cook them. Never eat any freshwater fish or shellfish unless it's been cooked properly. And I'm not going to explain to you why in any detail, but there are a whole bunch of really horrific parasitic diseases that you can get from eating raw freshwater shellfish and fish. So I think they're disgusting. They smell terrible. We handled them for almost 30 years in the lab at the Carey Institute. Never once did anybody stick their head in the labs and say, could I take some of those home when you're done? What they said, they would stick their head in the lab and they would pinch their nose and they would say, could you close the door? Now, what eats them? There's quite a few things that eat them, mostly this sort of specialized predators that can crush shells. So crayfish eat them, blue crabs eat them. Some of the fish that eat them are sturgeon eat a lot of them. And that one of the new invaders to the Hudson, the freshwater drum, probably actually followed zebra mussels into the river because they eat a lot of zebra mussels. And they've got teeth about like human teeth down in their throat. They can crush shells very readily. So there's a bunch of fish that eat them. Some of the diving ducks eat them.
Okay. A follow-up question regarding zebra mussels. Carol asks, where did zebra mussels come from?
So zebra mussels are native to southeastern Europe in the region of the Black and Caspian Seas and the Aral Sea. They got out of that area when Europeans built a whole bunch of canals linking those water systems to other water systems in the early 19th century. And then they came into North America, into Lake Erie, probably Port of Detroit, actually, in the mid-1980s in ballast water. Zebra mussels have little tiny, incredibly numerous planktonic larvae. So anytime you take raw water out of a place where zebra mussels live, like a bucket of water from the Hudson, it's going to be full of zebra mussel larvae. And so an ocean-going vessel dumped a whole bunch of larvae into the Port of Detroit, probably in the mid-1980s. And then they leapfrogged over into the Hudson. First detection 1991 near Catskill. Because they came into the middle of the river, they probably came in on the hull of a boat or a barge or something like that took them right into the middle of the river.
Which actually suggests an answer to the next question. They came in the middle of the river instead of at the north by the canals. And there's another question that does ask from M. Henkin, how have the invasives come to the Hudson? Well, there are obviously lots of different ways, but you might want to talk about canals and one or two other ways that they do get here.
Yes, that's actually something we know a fair amount about. And so in general, the pathways that invaders use vary over time because they depend on what human activities are important in the watershed or in the river. So in the 19th century, we had a lot of plants coming in solid ballast. So when ships used to ballast themselves, they would just take on stones and dirt and stuff like that, and there would be weed seeds mixed with that. We moved this to liquid ballast, ballast water, more recently, and so now we're seeing invaders like fish and invertebrates and phytoplankton, and some viruses and bacteria as well coming in ballast water. That's you pick up the water in the port of Amsterdam and then you dump it out in the port of New York. And so ballast water is important. The pet and horticulture trades have been important and are still important. You can buy a lot of things that, in my opinion, you should not be allowed to buy. And people release them into the wild when they get, tired of them. And then there's the canal. And so the Hudson's connected to other systems through currently, I think just two canals active. Steve, you may know if that's right, the Champlain and the Erie. In the past, there were additional canals as well. And so organisms can either go through their own power on those canals, or they can ride on a boat hull through those canals. So actually, the full story of the zebra mussel is there were two invasions of the Hudson. The first was there was an appearance in 1991 at near the village of Catskill, probably representing a boat or barge hull. And in 1992, they came to the Mohawk through the canal system. So in that case, we have two sources.
Interesting. One or two more questions on invasives, and then we'll move on to some other topics. Are there any, this is from Valerie, are there any viable control strategies for managing water chestnut? Or by extension, perhaps other invasives once they get here?
Yeah. So water chestnut, I'm not a real expert on this, but I'll tell you what little bit I do know. So when the water chestnut infestation is small, like sometimes people at a lake will find a few water chestnuts growing near the boat launch, where they've obviously come in on somebody's trailer or something like that. And there might be like 9 plants there. If you act quickly, you can pull them out. The trouble is once they get established, I guess a lot of you know this, the seeds can live for many years in the sediments. And so once they're well established, then you've got a long range control strategy. And I would encourage people in that case to be thinking about not eradication, but management. So like in the Hudson, they tried eradicating them in the Hudson. They sprayed huge amounts of 2,4-D onto the Hudson for I think about 15 years and failed to eradicate. That's Agent Orange. And it didn't eradicate them. The problem with water chestnut is that you can't get through it a lot of times, right? And so I know they're using weed cutters in some places in the Hudson, and that's a perfectly good strategy if you just want to cut a channel through a water chestnut bed so you can get kayaks and small craft, through the bed. We even played around. We never got funded for this, to do an experiment to see if we could increase oxygen levels in a water chestnut bed, not by eliminating the bed, but by just cutting channels through it. So there are some options depending on what stage the infestation is and what your goals are for management. And I really, really strongly encourage people, if you have a non-native species you're thinking about controlling, to ask yourself, why am I controlling it? And the answer, it's not native, is not, in my opinion, a good enough answer. You want to be thinking this species is tough on native fish or valuable fish that I like. It's ugly. It keeps me from getting my boat through. I think if you can identify specific management objectives, you're much more likely to come up with a satisfactory control.
One more and one last invasive question, then we'll move on. Gareth asks, are there any systematic searches going on in the Hudson Valleys or in the Hudson's tributaries for hydrilla, which was found in the Croton River system some years ago and is the object of a control project there?
I don't know. I haven't been in the Hudson Valley now myself in 3 or 4 years, and I'm out of the loop. There have been in the past some systematic searches to see how far it was spreading. And if someone on the call knows the answer to that, perhaps you could answer in chat and inform all of us. Are there systematic searches for hydrilla?
Yeah. Okay, we'll move on a bit. We have a question here from Bridget asking, how could you tell that the tomcod had evolved and that it wasn't just a lessening of whatever it was that caused the liver cancer?
Okay, this is kind of beyond what I know a lot about because I'm not a biochemist or a geneticist. Ike Wergen has done some really beautiful, detailed work on this, and there are several things that they know. They know that the gene frequencies in the Hudson River tomcod population are vastly different than the gene frequencies in other nearby tomcod populations. And that was the graph I tried to show you. So the Hudson tomcod are genetically distinct in a particular way. They know that tomcod carrying that gene variant are much less sensitive, about 100-fold less sensitive to dioxin and PCBs than the ones carrying the other form of the gene. The part that I couldn't find out about, and I don't know about, and I don't think has been done, is whether the incidence of liver cancers is declining in the Hudson River as a result of this evolution. And Ike has made this interesting case that it's possible that the tomcod can become less sensitive to the pollutant without reducing the incidence of liver cancer. It would be advantageous for the tomcod to be less sensitive to the pollutants because the pollutants also affect the baby survival of the baby tomcod and things like that. But tomcod spawn by the time they're 1 year old, and the cancers don't usually come in until their second year. And so it's possible that the liver cancers from a population point of view aren't threatening the tomcod as much as the survival of the young tomcod would be, if that makes sense.
Yeah, I seem to remember, and I'd want to check this before putting it out there, but I seem to remember that that as part of the liver cancer story, the population of tomcod skewed young. There were very
Oh,
few
yes.
fish older than 1 year, and that has changed a bit. We're starting to see a higher proportion of the older fish. I'd want to check that, but that's my memory about that.
Yeah, and that's exactly what you'd hope to see.
Yeah. On to a few of the questions about dealing with climate change here. And I'll start with a big one. This is from Sarah. What do you think of the proposal to create giant gates at the foot of the Hudson to guard against flood damage from storm surge?
I think you'd want to be really careful. So the proposal is that something like, and again, I'm not up on this completely, but it's something like to have giant gates at the mouth, somewhere around the mouth of the Hudson that could be closed in anticipation of a storm surge. And that such things exist in the Netherlands in some places. Depending on how they're built and how they're deployed, such gates could have large and unpredictable effects on the ecology of the area, the river and the area around the river. And so this is something that, I made some comment about hastily. I would hate to see something like that built in a big hurry. It's going to be really complicated, both from an engineering point of view and from an ecological point of view. And it would be something that you'd really want to put your best minds to for a while, not 10 minutes.
Another climate change question from Peter. Is the Hudson becoming more acidic with climate change?
Okay, so the reason Peter might think that would occur is that the rising CO2 in the atmosphere could make water more acidic. CO2, when it dissolves in water, forms a weak acid, carbonic acid, and that's what people are talking about in the ocean, right? And there's a lot of concern about the pH of the ocean, about the ocean becoming more acidic and therefore less suitable for things like corals, which are already having a terrible time of it. I can give two answers to Peter's question. The first is it's less likely to be an issue in the Hudson because the CO2 levels in the Hudson are not atmospheric CO2 levels. I showed you a graph of that, which most of you probably spaced out on, but the CO2 concentrations in the river are much, much higher than atmospheric, and they're controlled by respiration rates in the river. So it's not the rise in CO2 in the atmosphere wouldn't necessarily drive rise in CO2 in the water in the same way it does in the ocean. The second thing is that empirically we have a long enough record of CO2 in the river, is that it's been, or sorry, of pH in the river, that pH has been rising in the river a little bit over the last few decades. Um, and and so the river's been getting a little less acidic in terms of pH in the last few decades. And we think this is because of the chemistry of rain and snowfall interacting with the soils in the watershed.
Okay. Um, another question about impacts on the river, not necessarily from climate change, but we had a question about the proposed installation of the Champlain Express. Those are the power cables that are
Yeah.
proposed to be buried in the bed of the Hudson to deliver hydroelectric power from Canada to New York City. If the Hudson, and this is from Diane, if the Hudson is dredged in order to keep the shipping channel clear, would additional dredging to carry energy to New York City pose additional risk to the Hudson?
Yeah, it's a good question. I'm not sure I can answer it. I did read the impact materials for that project, gosh, 5 or 10 years ago, probably 10 years ago, when it was proposed, and they were arguing that that the effects of laying that cable on the bottom of the river would not be too large. And I think that for a lot of the length of the river, that's probably true. There are some places where it might be, the impacts might be a little greater. I don't remember enough from my reading of the document to be able to give you much of an answer, except that it was that problem was raised and addressed in the environmental impact statement.
Okay. Well, not unexpectedly, Dave, we have some fish questions here.
Okay.
Um, and I'll start with one that actually probably pertains to your expertise in zebra mussel ecology to some extent, and that is what is the latest view on why the shad population is declining in the Hudson? There's been various theories, including impact of zebra mussel on food chains.
Yeah, so I think that there's kind of two questions here, if I can take the liberty of. The first is, why did they decline? And the second is, why haven't they been recovering very well since the fishery was closed? In my opinion, the most likely explanation to why they declined was that they've been impacted by a whole bunch of different things, each of which has harmed the ability of the shad to reproduce and thrive in the river. So it happened a long time ago, but we lost 4,000 acres of river shallows in the upper part of the estuary that would have been prime rearing habitat for young shad. We still have harvests of shad offshore, and historically we had harvests of shad in the river that were pretty large. My group did some work on the zebra mussel invasion, and I believe that we found evidence that the zebra mussels have reduced the abundance and growth rates of shad and other plankton eating fish in the river. So the zebra mussels weren't good for them. And then you've got everything else that's happened to the river, right? The changing climate, the introduction of more and more exotic predatory fishes like channel catfish, the pollutants in the river, the history of pollutants in the river. And so I think that each one of these changes has probably made the shad population a little less resilient, a little less elastic, a little less able to recover from impacts. I asked the question about how they're doing now to DEC fisheries biologists earlier this week, because I thought I might be getting this question. And they think that one of the reasons that why the recovery has been slow is that there's still an unacceptable amount of mortality occurring offshore. And these are called bycatch fisheries, I think, where people fishermen aren't particularly trying to catch shad. They're trying to catch something else, but they catch and kill shad in the process of getting those other target species. And the DC folks think that these offshore fisheries are still too large to be good for the Hudson River shad stocks. But I come back to, I think if we had those 4,000 acres of shallows back, I think it's likely that the offshore fisheries would be less of an issue.
They could bounce back if we were able to control that.
Yeah.
Yeah, one has to remember, and I like to make this point, and I'll jump off that to say that we think of these as being our shad, but in fact, they migrate and spend most of their lives offshore, migrating from up to the Bay of Fundy and down to North Carolina and that area, and are impacted by fisheries and by conditions out in the ocean, perhaps even more than they are in the Hudson. So it's a complicated issue. A few other fish questions here. And one of them a non-fish question. It was bundled together with a fish one from Diane and Brian. Are oysters or sturgeon returning to the Hudson? Now, I'd be interested in the oyster, since you're an expert on mollusks. I'd be interested in your thoughts about the oyster restoration and about and I'll put in a question of my own here, make it more complicated. One of the rationales for oyster restoration is that they would have the ability to clean the harbor to some extent, given their filtering capacity. So if you could comment on that too. Are they being restored? Are they coming back? What is the potential for them to actually clean the water?
Yeah, so I don't know. In case some of you on this call don't know, there used to be immense beds of oysters around New York City, hundreds of square miles of them. In that native state, they almost certainly had an impact on water quality. They provided a lot of physical habitat. A lot of animals, invertebrates, and fish live in and around oyster beds, and they must have been a dominant factor in the ecology of the area around New York City. And there have been, in the last maybe 10 years, Steve, 15 years, there have been serious attempts to restore oyster beds around New York City again. And it's interesting because they're very explicit about this is not to provide oysters to eat.
Right.
In the old days, it's hard to imagine how many oysters were eaten that came out of New York Harbor and surrounding areas. But the restoration is not to provide oysters to eat; it's to provide oysters to filter water and to provide physical habitat for other animals in the river. And I've just been out of the loop the last few years too much to be able to tell you how well that's going. If somebody on the call wants to drop a note in chat, that would be great.
Yeah.
The sturgeon, the Atlantic sturgeon, at least, which is the larger of the two species in the Hudson, there's some evidence that, and I think, Steve, you may have mentioned this the other day, that we're starting to see more young sturgeon in the last 5 years or so in the river. And there's a 40-year moratorium, I think, on the fishery. And the intention is that if we're just patient, those fish will come back into the river, which would be awesome. And the sort of early returns here are looking promising, like that we may be seeing a little uptick in juvenile Atlantic sturgeon in the river.
Yeah. And a question that came up last week too, and we didn't have an answer for it, and I'm not sure you will either, but again, the question was raised about the numbers of white perch in the river, whether they're going up or down. And I don't have any idea about that one. Have you heard any research?
I didn't ask my DC
yeah.
folks. DEC has some information on white perch. I mentioned that some of the monitoring programs are disappearing. There were large fisheries monitoring programs associated with the electric generating utilities in the river that have gone away. And so we now have less information about fish populations in the river than we did a few years ago. And there is an attempt to restore some of that monitoring right now, which would be really good if we could do it.
Yeah. It's getting pretty late, so I'm gonna. I think we're gonna have to. The questions are still coming in, but I'm gonna say maybe one more here. And let's see. There was. We had a from Peter a question about proposals to put floating plant beds on the river, and I think he qualified that to say beds of cattails and things like that. Uh, I don't know. I'm not familiar with those proposals. Are those uh, anything that you're familiar with?
I've never heard about that in the Hudson. I've seen that uh, in sort of wastewater treatment systems where people will have a sort of smallish ponds, that they'll run the sewage effluent through a series of ponds to clean up the water before it's discharged into surface waters. And so sometimes they'll build these floating islands of things like cattails as part of that process to sort of more aggressively take pollutants out of the water before it's discharged. I don't know if that's what they're talking about or not. There would be really some logistical issues in the high energy Hudson with something like that.
Yeah, yeah. It was specific. The question was about floating plant beds to replace loss of cattail marshes. Um, all right. One last question, and then we'll uh, let you go. Um, that is a question about the historical levels of marine mammals in the Hudson. North of New York City, there are occasional seals, but were they ever abundant?
And I think that question's for you, Steve. You're
All right, I think I had to
I
write
don't
about
know
that.
anything about that.
I actually wrote about that in the book. Um, yeah, actually seals seem to be increasing in abundance, and everybody's heard about I would hope about the increase of great whales and actually dolphins and other smaller cetaceans off the port of New York. That's largely linked to the increase, scientists think, in the numbers of menhaden, a fish that's commonly called bunker, which is a really important forage fish for many, many species, including the whales. Um, and they do come up the river. The menhaden come up the river. Dave mentioned the studies showing how fish don't always do what we expect. And I think I commented last time that young bunker do come into the Hudson in their first year, and it turns out that a lot of them spend the winter in the Hudson and are still here the following year. But and it's I'll point out that management of that species has recently been adjusted to take into consideration their ecological importance. They are such an important forage fish that the National Marine Fisheries and the Atlantic State Marine Fisheries Consortium Council are starting to weigh the impacts of or the importance of these fish to marine food chains, as well as their importance to commercial fishing. We don't eat menhaden directly. They are used for animal feeds, for fish oils, and things like this. And it is one of the largest commercial fisheries on the East Coast. So the fact that, and Dave mentioned this in his talk, wise management, informed management of science-informed management is going to be critical to facing issues in the future. And I think I'd like to think this is an example of enlightened fisheries management where they're really protecting the species for ecological purposes as well as just maintaining it so we can catch more of them.
So, Steve, I see that John Lipscomb has answered the oyster question for us.
Okay, John.
So people who are interested in how the oyster restoration is going can Google Billion Oyster Project to get an entree into that project. Thank you, John. I guess I did one more soapbox thing I'd say about oysters. Don't do it yourself, okay? Oyster restoration projects around the world have introduced a lot of bad non-native species, right? Because people will just take a bucket of oysters from one place and take it to another place. And when they do that, they take a bucket of a whole bunch of other species with the oysters. So well-managed oyster restoration projects can be a really good thing. Amateurs doing it you can be the guy that brings the next terrible species into the Hudson, and you don't want to be that guy.
Okay, with that, I think we're going to sign off now. But Dave, really a wonderful, wonderful lecture, and I'd like to thank you and also all the presenters for the webinar series. This being the last of them, Brian Forrest and Roger Panetta as well. We really appreciate you spending the time preparing this and even talking to the fisheries biologists so you'd be ready for some of these questions. So thank you to all who have signed on, and Clearwater will certainly be considering doing more such webinars in the future, and we will keep you posted about that. Thanks again for signing on. Thanks to Dave, and I wish you all a good night.
Good night.